There's something about blueberries that makes them seem much simpler than they really are. Perhaps it is their size, or the quiet familiarity of finding them scattered across a bowl. Yet beneath that dusty blue surface is a story shaped by acidic soil, vibrating bees, microscopic wax, and pigments that change as they pass through the human body. Whether you have returned tonight or are discovering this channel for the first time, this is a quiet place to slow down, learn something gentle, and allow the day to gradually fade into the background. A cultivated blueberry commonly weighs only 1 or 2 grams and is small enough to rest on the tip of a finger. Its surface appears dusty blue. Its shape is almost round, and one end carries a tiny 5-pointed crown. But those visible details come from several different parts of the fruit. The pale coating is a natural layer of wax called the bloom. It is not dirt, and it is not something added after harvest. If the bloom is rubbed away, the skin underneath appears much darker, often closer to purple-black than blue. Cut the same berry open, and another surprise appears. The inside is usually pale green or nearly colorless. Most of the strong red-purple pigment is concentrated in the thin outer skin. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. Even the little crown has a specific origin. It is made from the dried remains of the flower that produced the fruit. Several months earlier, those pointed pieces surrounded a small bell-shaped blossom growing on a woody shrub. The berries slowly formed behind that flower after pollen reached its center, and tiny seeds began developing inside. Every ripe blueberry carries this record of its earlier life. Its crown remembers the flower. Its seeds record successful pollination. Its waxy surface protects the skin while changing the way light reflects from it. Its pigments mark the moment when the fruit became ripe enough for an animal to eat. Those pigments are known as anthocyanins. They belong to a much larger family of plant compounds, and they are responsible for much of the blueberry's reputation as an antioxidant-rich food. But the blueberry plant did not make anthocyanins for a person's breakfast. It produced them as part of its own growth, protection, and reproductive cycle. The amount that develops in one berry can be influenced by the plant's genes, the hours of sunlight it receives, the temperature during ripening, and exactly when it is picked. Long before any color appears, the bush must survive in soil far more acidic than most garden plants prefer. Its shallow roots work alongside microscopic fungi that help collect nutrients from decaying organic matter. The bush must then pass through enough winter cold for its flower buds to open normally in spring. When those flowers appear, their pollen remains trapped inside narrow structures that certain bees must vibrate to empty. Only after successful pollination can the green fruit begin its slow change towards sweetness, softness, and dark pigment. Harvesting does not bring those changes to a complete stop. The berry remains living tissue after it leaves the bush. It continues using oxygen and releasing carbon dioxide as its stored sugars are gradually consumed. Cooling slows this process, which is why fresh blueberries are moved into refrigeration soon after harvest. Another transformation begins when the berry is eaten. Chewing breaks the skin and releases its water, sugars, acids, fiber, and pigments. Stomach acid changes the form of some anthocyanins. The small intestine absorbs only part of what arrives. Farther along, gut microbes break other compounds into smaller pieces that may later enter the bloodstream. By the time anything reaches a blood vessel, liver cell, or brain, much of the original berry chemistry has already been rebuilt into new forms. This is why a blueberry cannot be understood through a single laboratory score or a simple claim about antioxidants. Its story begins in soil, passes through fungi, winter buds, flowers, bees, fruit cells, and reflected light, and then continues through an entirely different set of systems inside the body. To follow those changes clearly, we first need to understand what kind of fruit a blueberry actually is. Botanists place blueberries in a genus called Vaccinium, pronounced vac-sin-e-um. A genus is a group of closely related species that share important inherited features. More than 400 Vaccinium species grow across the world, ranging from low ground-hugging plants to woody shrubs several meters tall. Cranberries belong to this genus, along with bilberries and the red lingonberries that grow across northern forests and tundra. Most commercial blueberries belong to a smaller group traditionally called Cyanococcus. This group includes several species native to North America. Northern highbush blueberry carries the scientific name Vaccinium corymbosum. The common lowbush blueberry is Vaccinium angustifolium. Rabbit-eye blueberry, a taller plant adapted to the southeastern United States, is usually called Vaccinium virgatum. All three may produce fruit sold as blueberries, yet their bushes differ in height, cold tolerance, growing season, and the chemistry inside their fruit. The everyday name therefore describes a family resemblance rather than one globally identical plant. Despite that variety, blueberries share a basic fruit structure. Botanically, they qualify as true berries. A true berry develops from one flower containing one ovary. The ovary is the part of the flower that holds the ovules, which can become seeds after fertilization. As a blueberry develops, the wall of that ovary thickens and becomes the soft fruit surrounding those seeds. This creates a continuous layer of edible flesh, rather than a group of smaller fruits pressed together. Strawberries follow a different plan. Their red flesh develops mainly from the flower's swollen base, known as the receptacle. Each pale speck on the surface is a separate dry fruit called an achene, with a seed held inside it. Raspberries develop from many ovaries within one flower. Each rounded piece of a raspberry is a tiny fruit called a drupelet, and the collection of drupelets forms the berry-shaped object we eat. Ordinary language groups all of these fruits together because they are small, soft, and often sweet. Botanical language separates them according to how their flowers become fruit. A blueberry fits the formal definition more closely than either a strawberry or a raspberry. Its construction becomes clearer when the fruit is examined from the outside inward. The outermost covering is the cuticle, a thin protective layer produced by skin cells. Wax crystals collect on this cuticle and form the pale blooms seen on fresh berries. Beneath the wax lies the epidermis, the outer layer of cells that makes up the skin. Most cultivated blueberries store their strongest anthocyanin pigments within and just below this surface. The skin also contains a concentrated share of the fruit's fiber. Below it lies the pale pulp formed from the ovary wall. Water fills most of this tissue, while dissolved sugars and organic acids help create the berry's flavor. Fine vascular strands once carried water, minerals, and sugars into the developing fruit. These strands entered through the pedicel, the narrow stalk connecting the berry to its branch. When a ripe berry is picked, it separates from that stalk and leaves a small circular scar. A clean, dry scar helps a harvested blueberry resist moisture loss and decay. At the opposite end sits the familiar crown. Its pointed shape comes from the persistent calyx, the small leaf-like parts that once rested around the flower. Those pieces remain attached while the ovary behind them expands, leaving the flower's outline on the mature fruit. Inside the pulp are several small chambers that began as compartments within the ovary. Each chamber originally held ovules waiting to be fertilized. A well-pollinated blueberry can develop several dozen seeds, although many remain so small and soft that they pass almost unnoticed while the fruit is eaten. Some ovules never complete development. The number of successful seeds still matters, because growing seeds release chemical signals that encourage nearby fruit cells to divide and expand. Better seed development can therefore contribute to a larger berry, and, in many cultivars, earlier ripening. Every visible layer will play a different role as the journey continues. Skin concentrates pigment and fiber, while wax changes the light returning from the surface. Seeds influence the growth of the flesh surrounding them. These shared structures developed within a group of plants shaped by acidic wetlands, cold forest margins, and open ground across North America. Across North America, that shared fruit structure developed in plants with very different shapes. Northern highbush blueberry, Vaccinium corymbosum, grows naturally along wet forest edges, around bogs, and in other moist places with acidic soil. A mature highbush plant commonly reaches between 6 and 10 feet tall, placing many of its berries near eye level. Its natural range extends through much of eastern North America, from the southeastern United States into southern Canada. Farther north and closer to the ground, Vaccinium angustifolium follows another growth plan. This is the common lowbush blueberry found across northeastern states, the Canadian Maritimes, and parts of Quebec and Ontario. Most lowbush plants remain between 6 and 24 inches tall. Their branches can disappear beneath snow during winter, where the surrounding snow helps protect them from colder air above. Commercial blueberry farming eventually carried North American species to other continents. The wild plants that began that expansion were already adapted to wetlands, rocky barrens, sandy clearings, and the edges of northern forests. A lowbush field also contains far more below-ground structure than its short stems suggest. Horizontal stems called rhizomes spread through the upper soil. New shoots rise from buds along each rhizome, allowing one connected plant to cover a patch several feet across. A patch may contain hundreds of stems, while remaining one genetic individual. The neighboring patch can belong to a different clone with its own inherited traits. During autumn, one clone may turn bright red, while another remains green for several extra days. Flowering dates, berry size, sweetness, and ripening time can also change at the boundary between patches. From a distance, the field looks continuous. Closer inspection reveals a living mosaic assembled from many plants spreading beside one another. This low growth works especially well in places that are disturbed repeatedly. When a storm opens the forest canopy, additional sunlight reaches shrubs close to the ground. Fire can remove taller competitors, while underground rhizomes survive beneath the heated surface. Cutting, grazing, and cleared land can produce similar openings. Ecologists call this early successional habitat. It is the bright stage that develops after disturbance, before young trees grow tall enough to close the canopy again. Blueberry leaves require that sunlight to manufacture the sugars later moved into flowers and fruit. As shade increases, berry production usually declines. The wild blueberry barrens of Maine and eastern Canada are managed versions of this older ecological pattern. Growers often divide their land into two groups and place each group on a 2-year cycle. After harvest, one part may be mowed close to the soil or treated with a carefully managed burn. Fresh stems grow during the following summer and form buds near the end of that season. Those stems flower and produce berries during the second year. While one part of the field is producing fruit, another part is rebuilding its stems. The cycle creates the open, sunny conditions that low-bush blueberries have used for thousands of years. Animals have helped the plants move through those landscapes. American robins and cedar waxwings swallow ripe fruit and later release some of the seeds in their droppings. Black bears can consume large quantities during late summer, carrying seeds much farther than a small bird might travel between meals. A seed that lands in suitable acidic ground may begin a new genetic individual, while established plants continue spreading through rhizomes. People entered this relationship long before the first commercial cultivars appeared. Wabanaki peoples of Maine and the Canadian Maritimes gathered wild blueberries when the barrens ripened in summer. Communities including the Passamaquoddy and Mi'kmaq ate berries fresh, dried them for storage, and cooked them with other foods. These harvests belonged to specific local traditions, formed many generations before deliberate blueberry breeding began in the early 20th century. The blueberry's present range therefore carries traces of underground growth, animal movement, repeated disturbance, and long human use. Open sunlight explains where the plants can flourish above ground. Their survival still depends on what happens within a shallow layer of unusually acidic soil. Just beneath a low-bush patch, most of the working roots occupy the upper several inches of earth. This layer receives fallen leaves, small pieces of bark, and other plant material that slowly decompose. It also contains the acidic conditions preferred by nearly every commercially important blueberry. Most blueberry bushes grow best when soil pH remains around 4.5 to 5.2. The pH scale measures the concentration of hydrogen ions in a solution. 7 represents a neutral value. Each whole step on the scale marks a tenfold change. So soil at pH 5 contains about 10 times the hydrogen ion concentration of soil at pH 6. Compared with neutral soil at pH 7, the difference reaches about 100 times. That chemistry changes how nutrients dissolve and how easily roots can absorb them. Blueberry roots are extremely fine and often appear as a dense brown network mixed through organic material. Their active roots may be thinner than a piece of sewing thread. They also possess few effective root hairs. On crops such as wheat, thousands of tiny root hairs extend from the root surface and increase the area available for absorbing water and minerals. A blueberry has much less of this added surface. Its shallow roots can reach freshly decomposing material near the soil surface, although they dry quickly when heat removes moisture from that layer. Even a mature bush can suffer during drought because most of its absorbing system remains close to the surface. Part of the missing reach is supplied by fungi. Blueberries form a partnership called ericoid mycorrhiza, named for the heath family of plants to which blueberries belong. Mycorrhizal fungi produce microscopic threads known as hyphae. These threads spread through spaces between soil particles and enter the outer cells of the finest blueberry roots. A hypha can explore pores too small for the root itself to enter. Fungal enzymes also help release nitrogen from complex organic material that the plant cannot easily break apart on its own. Some of the recovered nitrogen, phosphorus, and other nutrients move toward the root. Carbon compounds made in the blueberry's leaves travel in the opposite direction and feed the fungus. The exchange begins with photosynthesis above ground. Leaf cells use sunlight to combine water with carbon dioxide, producing sugars that can be transported through the plant. A portion eventually reaches the root and its fungal partner. This relationship helps blueberries occupy soils where nutrients may be present, while remaining chemically difficult to obtain. Nitrogen offers a clear example. Plants absorb much of their nitrogen in two common forms: ammonium and nitrate. Blueberries are especially adapted to ammonium, the form often found in acidic organic soils. Their ability to use large amounts of nitrate is more limited than it is in many field crops. Soil acidity also changes the behavior of metals. When pH rises too high, iron becomes difficult for blueberry roots to absorb. Young leaves can then develop iron chlorosis, a condition in which tissue turns yellow while the veins remain greener. Extremely low pH can make manganese and aluminum more soluble, allowing them to reach harmful concentrations. Water creates another narrow balance. Blueberries often grow near wetlands and in moist ground, yet their roots still require oxygen. Water filling every airspace can slow root respiration and damage the absorbing tissue. Good blueberry soil holds steady moisture while allowing excess water to drain away. This combination explains why a bog edge can support healthy bushes, while a permanently flooded hole nearby cannot. Acidity reduces competition from many plants. Fungal threads improve access to organic nutrients, and shallow roots use the rich surface layer. Together, these conditions create a narrow chemical home. Throughout summer, the partnership supplies materials used to build leaves and extend stems. Some of the resulting sugars remain stored after growth slows, because the buds for a future crop begin forming before the current year has ended. By late summer, a blueberry stem has already begun preparing flowers for the following spring. Small buds form where leaves join the woody shoot. Narrow, pointed buds usually contain future leaves and stem growth. Flower buds become rounder and fuller as several miniature blossoms develop inside them. One high-bush flower bud may hold roughly 5 to 10 individual flowers. Their petals, pollen structures, and young ovaries begin forming while ripe berries from the current season may still remain on nearby branches. Producing those buds requires energy collected months before they open. Leaves manufacture sugars during summer and send part of that supply into stems and roots. Starch provides one form of long-term storage. When growth begins again, stored carbohydrates help open buds and support flowers before the new leaves have reached full size. The approach of winter changes both the visible bush and the living cells inside it. Shorter daylight helps slow shoot growth. Cooling temperatures encourage a process called cold acclimation, which gradually increases the plant's ability to survive freezing. Cell membranes change their mixture of fats so they remain flexible at lower temperatures. Dissolved sugars become more concentrated within the tissues and help protect membranes and proteins. Special protective proteins can reduce damage as water freezes in spaces outside the cells. Leaves continue sending useful materials back into the plant before their connections finally seal. Red anthocyanin pigments often become visible as green chlorophyll breaks down. The leaves eventually fall, leaving closed buds attached to bare stems. Dormancy keeps those buds from responding to every brief warm period at the beginning of winter. Many blueberry cultivars must first receive a certain amount of cool weather. Growers describe this need through chilling hours or more detailed chilling units. A simple chilling hour model counts time below about 45 degrees Fahrenheit or 7 degrees Celsius, although modern models give different temperatures, different values. The exact requirement depends on the cultivar. Some southern high-bush blueberries need only a few hundred hours of suitable cold. Many northern high-bush cultivars require roughly 800 hours or more. A northern plant moved to a very warm winter climate may leaf out unevenly, flower poorly, or keep some buds closed. A low-chill plant grown far north faces a different timing problem. Once its smaller cold requirement has been met, an unusually warm period can encourage development too early. Completion of chilling removes an internal block that helped hold the bud closed. Later warmth then controls how quickly the prepared tissues expand. This sequence makes the order of temperatures especially important. A fully dormant bud can survive air far colder than an exposed flower. As the bud swells and green tissue becomes visible, some of that freezing tolerance disappears. Open high-bush blossoms may be injured near 28 degrees Fahrenheit, or about -2 degrees Celsius. The exact damage depends on the cultivar, the length of the freeze, and the blossom's stage of development. A warm February followed by a hard March frost can therefore cause more crop damage than steady cold that keeps the buds closed. Breeders must match this seasonal clock to the growing region. Cultivars used in Ontario or Maine need enough cold tolerance for long winters and enough chilling to remain synchronized with spring. Bushes grown in Georgia or Florida benefit from lower chilling requirements because their winters provide fewer cold hours. Weather can still shift the result from year to year, even when the cultivar normally suits the region. Inside each closed flower bud, the future crop waits in a partly formed state. Stored sugars remain available in the wood below it. After sufficient chilling and warmth, the bud scales separate and reveal pale blossoms, whose downward-facing shape keeps their pollen unusually well contained. As the bud scales separate, several pale flowers begin extending from the same rounded bud. Their petals are usually white or soft pink and fuse together into the shape of a narrow bell. Each flower hangs with its opening pointed toward the ground. This position helps shelter its reproductive parts from rain, although it also places the pollen behind a physical barrier. Inside the flower are long structures called anthers. Anthers produce and hold pollen grains. On many familiar flowers, each anther splits along its side and leaves the pollen exposed. Blueberry anthers keep most of their pollen enclosed and release it through small openings near their tips. Botanists call these poricidal anthers because each opening acts like a tiny pore. A visiting insect must shake the dry pollen through those pores. Bumblebees are especially effective at operating this system. A bumblebee lands on the downward-facing flower and grips it with its legs. The bee then disconnects its wings from the powerful muscles normally used for flight. Those muscles contract rapidly, while the wings remain still. Vibrations travel from the bee's body into the flower and shake pollen from the anthers. The sound produced during this action is often higher and sharper than the hum of ordinary flight. Pollen may fall onto the bee's underside or collect around its legs before some of it is carried to another blossom. This process is known as buzz pollination. Tomatoes and several other plants use similar pollen release systems, though each flower responds best to a particular range of vibration. Honeybees can visit blueberry flowers and transfer pollen, but they cannot produce true buzz pollination. They often collect loose pollen or reach into flowers after another insect has shaken them. Their value depends partly on flower shape. A honeybee may struggle to reach nectar inside a long, narrow blueberry flower, while a bumblebee's longer tongue can reach farther into the opening. Field observations in Maine show that bumblebees may work roughly 10 to 20 lowbush flowers in 1 minute. Honeybees commonly visit about 5 to 9 during the same amount of time. One bumblebee visit can place more than 50 pollen grains on the stigma, the sticky surface that receives pollen. A typical honeybee visit in those observations deposited between 0 and 10. The difference can become important across a field containing millions of flowers. Maine lowbush blueberry fields have recorded 59 species of native bees. Some are small, solitary bees that nest in soil. Bumblebee queens emerge after surviving winter and begin searching for nest sites while blueberry plants are flowering. Body size, tongue length, cold tolerance, and movement between patches all influence how useful a species becomes to the crop. Cool spring mornings can keep honeybees inside their hives, while some native bees continue working. Growers may bring managed honeybee colonies into fields when wild pollinators cannot cover the full bloom. In productive fields, several hives may be placed on each acre. Increasing hive numbers can improve fruit set, although the result still depends on compatible plants being available. Many blueberry cultivars can use some of their own pollen. Pollen from another compatible cultivar often produces more successful seeds. That improvement can lead to larger berries, more even ripening, and a greater share of flowers becoming fruit. A bee moving between bushes therefore carries genetic material that will influence the crop weeks later. Once a pollen grain reaches the stigma, the flower has received the necessary material. Its progress still depends on a microscopic journey through the center of the flower toward one of the waiting ovules. Moisture on the stigma causes a compatible pollen grain to become active. The grain germinates and produces a narrow tube that enters the flower's style. This style connects the stigma above with the ovary below. A pollen tube grows by extending its tip through the soft tissue, guided by chemical signals released within the flower. Its width is measured in micrometers, with 1 micrometer equaling 1,000th of a millimeter. Many tubes may be growing through the same style at once. Each carries reproductive cells toward one ovule inside the ovary. When a tube reaches a compatible ovule, fertilization begins the development of a seed. The blueberry ovary sits below most of the flower's visible parts. This arrangement is called an inferior ovary, meaning the petals and sepals appear to attach above it. Sepals are the small leaf-like pieces that protected the unopened flower. Together, they form the calyx. After the petals fade and fall, the calyx stays attached to the far end of the enlarging ovary. Its pointed remains eventually become the crown on a ripe blueberry. The ovary contains several internal chambers. Each chamber holds multiple ovules, creating the possibility of several dozen seeds within one fruit. A mature blueberry may contain between 10 and 60 tiny seeds, depending on species, cultivar, and pollination success. Many remain small enough to pass unnoticed during eating. Developing seeds influence more than reproduction. They produce plant hormones that travel into the surrounding ovary wall. These signals encourage cells to divide and later enlarge. A flower that develops more viable seeds commonly produces a larger berry. In several cultivars, well-seeded fruit also ripens earlier than fruit with fewer successful seeds. This helps explain why poor pollination can create berries that remain small, even when the bush receives enough water and fertilizer. Contact with pollen does not guarantee a completed seed. The pollen must be genetically compatible with the flower. Some blueberry cultivars readily use their own pollen, while others perform better when pollen arrives from another cultivar blooming at the same time. Differences become especially visible in wild low-bush fields. Several vaccinium species may grow beside one another and open flowers during overlapping weeks. Pollen from sourtop blueberry, Vaccinium myrtilloides, can sometimes germinate on common low-bush blueberry. Fertilization may begin before the ovary aborts several days later. Those occupied ovules can no longer receive more compatible pollen. A field containing a high proportion of incompatible plants may therefore have limited fruit set even when many bees are present. Adding more hives increases the number of visits without changing the genetics of the pollen being carried. Successful fertilization begins a transfer of resources into the ovary. Sugars made in the leaves move toward the young fruit through vascular tissue. Water and minerals arrive from the roots. The petals have completed their visible role. Yet the crown remains attached while the ovary slowly expands around its developing seeds. For several weeks, the result is a firm green sphere that gives little sign of the blue fruit it will become. Soon after the petals fall, cells inside the young ovary begin dividing rapidly. Each division produces more cells that can enlarge later. This early period helps establish the berry's eventual capacity for growth. Developing seeds draw sugars, minerals, and other materials through fine vascular strands connected to the fruit stalk. The surrounding flesh remains dense and firm. Its green surface contains chlorophyll, the same light-absorbing pigment found in leaves. Although the small fruit receives most of its energy from nearby leaves, its skin can perform a limited amount of photosynthesis. Sunlight helps turn carbon dioxide and water into sugars within those green cells. Young blueberries also contain high levels of organic acids. Citric acid is usually the most abundant, with smaller amounts of malic and quinic acids contributing to the sharp flavor. Sugar concentration remains low enough that an immature berry tastes strongly sour. Firm cell walls and pectin hold the tissue together. Pectin is a group of complex carbohydrates that helps bind neighboring plant cells. Blueberry growth does not proceed at one steady speed. Researchers often describe its development with a double sigmoid curve. A sigmoid curve begins slowly, rises quickly, and then levels. The first rise follows early cell division and enlargement. Growth slows during a middle interval, while seeds and embryos continue developing. A second rise occurs as ripening begins and fruit cells take in additional water. The exact timing changes with cultivar and temperature. Some early ripening blueberries move from flowering to harvest in roughly 6 weeks. Later cultivars may require 10 weeks or more. Water reaches the fruit through vascular tissue and then crosses cell membranes. Dissolved sugars help draw water into expanding cells. Pressure builds against the skin as those cells enlarge. Steady moisture supports this process because blueberry roots remain close to the soil surface. Dry weather can reduce berry size when roots cannot replace water lost through leaves and fruit. Heavy rain near harvest creates the opposite pressure. A ripe berry can absorb enough water for its internal pressure to rise faster than the skin can stretch. Susceptible cultivars may split around the crown or picking scar. Conditions also vary within a single fruit cluster. The first flower in a cluster may open several days before the last. Bees may visit each blossom on a different morning, and fertilization adds another difference in timing. One branch can hold hard green berries beside fruit that has already turned pink. A few fully blue berries may be ready for harvest, while their neighbors remain a week behind. Handpickers return to the same bush several times, so each berry can remain attached until it is properly ripe. Mechanical harvest works best when a cultivar ripens more uniformly. During the green stage, the plant continues protecting its investment. Firmness discourages many animals from eating the fruit before its seeds are ready. Sourness provides another reason to leave it alone. Once seed development reaches a useful stage, that protective chemistry begins changing. The fruit expands again, while its skin starts sending a completely different visual signal. The first visible sign may be a faint patch of pink spreading across the green skin. Inside the fruit, several processes have begun changing together. Maturing seeds have reduced their demand for construction materials. The plant can now make the fruit more attractive to an animal capable of carrying those seeds elsewhere. Sugars continue arriving from nearby leaves. Glucose and fructose become the main simple sugars in a ripe blueberry, with smaller amounts of sucrose in many cultivars. Organic acids remain present, though sweetness becomes more noticeable as their concentration falls or becomes diluted by the berry's final growth. A berry's flavor depends on the ratio between sugars and acids. Two cultivars containing similar sugar levels can taste different when one retains more citric acid. Temperature and sunlight during ripening can change this balance. Cool nights may slow sugar use through respiration, while prolonged heat can speed development and reduce the time available for flavor to accumulate. Aroma also changes. Blueberries release small airborne molecules called volatile compounds. These include forms of linalool, geraniol, and several aldehydes that contribute floral or fresh green notes. Concentrations vary enough that one cultivar can smell noticeably different from another. At the same time, enzymes begin loosening the fruit structure. Some enzymes modify pectin between neighboring cells. Others change cellulose and related material in the cell wall. The cells remain filled with water, though their connections become easier to separate during chewing. Softening improves eating quality and makes the berry more vulnerable to bruising. A small impact during packing can damage internal cells without breaking the skin. Fungal spores later use those damaged areas as easier entry points. The most visible ripening change occurs in the skin. Chlorophyll breaks down and green color fades. Genes involved in the phenylpropanoid pathway become more active. A pathway is a connected sequence of chemical reactions, with each step controlled by an enzyme. This particular pathway begins with an amino acid called phenylalanine and eventually produces several families of plant compounds. One branch creates anthocyanins. Blueberry skin can contain pigments based on malvidin and delphinidin, along with forms derived from petunidin, cyanidin, and peonidin. Each base pigment can carry different sugar groups, producing more than a dozen related anthocyanins in one fruit. Their mixture changes the skin from pink to red-purple, and then, to a color so dark, it can appear almost black beneath the wax. Most cultivated high-bush blueberries keep these pigments close to the outer surface. The flesh remains pale. Bilberries, which are close relatives in the genus Vaccinium, often develop dark pigment throughout their flesh as well. Picking time controls how much of this process occurs on the bush. A berry may continue changing color after harvest, especially if it was nearly ripe. Its connection to the leaves has already been broken, so it cannot receive the same continued supply of sugar. Fruit picked too early may become softer and bluer, while retaining a flatter, more acidic flavor. Growers therefore judge ripeness through several traits, including surface color, firmness, taste, and how easily the berry separates from its stalk. By the time the fruit appears fully blue, its skin contains mainly red-purple pigments. The familiar blue must be produced by the way another layer handles incoming light. A freshly picked blueberry can reveal its color trick with one gentle touch. The untouched surface appears dusty blue, while a rubbed patch becomes smooth and almost black. Pressure from a fingertip removes part of the pale coating known as the bloom. Underneath it, the skin contains anthocyanins that appear dark red or purple. Crushing the berry makes the difference even clearer. Juice, spreading from the broken skin, looks violet, magenta, or deep red, according to its acidity and pigment mixture. No bright blue dye pours from the fruit. Much of the color that gave the blueberry its name comes from the way its surface handles light. The bloom begins as wax made by cells in the fruit's outer skin. Wax molecules travel through the cuticle, which is the thin protective layer covering the berry. Once they reach the air, those molecules solidify into structures far smaller than a grain of dust. Many form narrow tubes mixed together without a repeating pattern. Their size places them within the same tiny scale as wavelengths of visible light. Blue light has wavelengths of roughly 450 to 495 nanometers. 1 nanometer equals 1/1000th of a meter. Ultraviolet light occupies even shorter wavelengths, mostly below 400 nanometers. When sunlight reaches the bloom, its microscopic wax structures scatter a strong share of the blue and ultraviolet light back outward. The dark anthocyanin-rich skin beneath absorbs much of the remaining visible light. This dark background makes the pale blue reflection easier for an eye to detect. Color created partly through microscopic shape is called structural color. Pigment molecules produce color by absorbing particular wavelengths. Structural color appears when tiny physical features redirect light before it can disappear into the material below. Blueberry wax has a disordered arrangement, creating a soft, even blue that changes little as the viewing angle moves. Researchers examined this effect in detail in a study published in 2024. The work involved scientists at the University of Bristol and international collaborators. They studied high-bush blueberries and several other fruits with blue-looking waxy surfaces. A scanning electron microscope sent electrons across prepared samples and produced images of structures much smaller than ordinary light microscopes can resolve. Those images revealed tangled tubular shapes within the blueberry bloom. Instruments also measured which wavelengths the intact wax reflected most strongly. The reflected range extended through blue and into ultraviolet light. To test whether the fruit's surface structure could create the color on its own, the researchers removed wax and allowed it to crystallize again on a dark card. The new coating reflected blue and ultraviolet wavelengths, even though it was no longer attached to blueberry skin. This result showed that the wax layer could reproduce the visual effect away from the fruit. Related structures were found on plums and sloes, along with the blue berry-like cones of juniper plants. The bloom serves several practical functions while it changes the fruit's appearance. A waxy cuticle slows the movement of water from the juicy interior into dry surrounding air. Its surface also causes many water droplets to bead instead of spreading into a continuous film. Less standing moisture can make conditions less favorable for fungal spores attempting to germinate. Wax creates a physical boundary between living skin cells and dust or microbes arriving from outside. The protection remains incomplete, since ripe blueberries can still lose water, bruise, and develop mold. Handling gradually damages the delicate surface. Fingers, conveyor belts, and neighboring berries can flatten or remove the microscopic structures. A berry, with an even pale bloom, has usually experienced less rubbing than one covered with shiny dark patches. This is one reason growers and packers treat the coating as a sign of careful handling and freshness. The blue surface can also make ripe fruit visible against green leaves. Many fruit-eating birds detect blue wavelengths, and several species can see ultraviolet light that humans cannot. A reflected signal can therefore advertise mature seeds to an animal capable of carrying them away. The berry's familiar color emerges from a partnership between dark pigment and nearly invisible wax. Beneath that optical layer, the red-purple anthocyanins still perform chemistry shaped by the needs of the plant that produced them. Inside the blueberry skin, The red-purple color is held within individual living cells. Its pigments belong to a large family of plant compounds called flavonoids. The colored members of this family are known as anthocyanins. They dissolve in water, allowing them to remain suspended in the watery interior of a cell. Most are stored inside vacuoles, which are fluid-filled compartments surrounded by their own membranes. A vacuole can occupy most of the space within a mature skin cell. Concentrating pigment there separates it from many reactions occurring in the cell's working outer layer. In cultivated high-bush blueberries, pigment stays mainly in the epidermis and several cell layers beneath it. The pale flesh contains far less. Bilberries distribute anthocyanins through their pulp, which is why their juice stains more deeply. The color inside a vacuole depends partly on acidity. A strongly acidic environment favors redder molecular forms, while changes in pH can shift the same pigment toward purple. Nearby minerals and other flavonoids may alter the final shade. Attached sugars also affect how easily a pigment dissolves and how stable it remains. The colored central molecule is called an anthocyanidin before sugars are attached. Blueberries contain anthocyanins based on several anthocyanidins. Malvidin and delphinidin forms are often abundant in cultivated high-bush fruit. Petunidin, kyanidin, and peonidin add further variation to the mixture. A berry may contain more than a dozen individual anthocyanins because each central structure can carry different sugars. Blueberry cells manufacture these compounds from smaller starting materials. The pathway begins with phenylalanine, an amino acid also used to build proteins. An enzyme called phenylalanine ammonia lyase changes it into the first material in a broader pathway used for plant defense and structure. Several reactions later, chalcone synthase helps assemble a new ring-shaped compound. Additional enzymes alter that structure until anthocyanidin synthase produces the colored central framework. Other enzymes attach sugars and help transport the completed pigments into vacuoles. Each step depends on instructions carried by genes. Regulatory proteins can increase or reduce the activity of several pigment genes together. Their response helps explain why color develops quickly as a berry ripens. Light reaching the fruit can strengthen pigment production. Berries exposed to the sun sometimes accumulate more anthocyanin than heavily shaded berries on the same bush. Temperature changes the outcome as well. Cool conditions during part of ripening can support pigment formation in some cultivars, while prolonged heat may reduce the final concentration. Genetics sets another limit because each species and cultivar carries a different collection of enzyme instructions. The plant gains several possible advantages from this chemistry. Anthocyanins absorb portions of ultraviolet and visible light before that energy reaches sensitive structures deeper inside a cell. Young leaves sometimes produce red pigment during cold weather or intense sunlight. Damaged tissues may activate related pathways after injury or infection. Within plant cells, the pigments can react with unstable molecules formed during photosynthesis and stress. This antioxidant activity helps regulate chemical conditions in the tissue where the pigments are stored. Ripe fruit adds an ecological purpose. A dark berry stands out against green leaves and signals that its seeds have matured. American robins, cedar waxwings, and black bears can eat the fruit and move seeds away from the parent bush. The plant supplies water and sugar during an exchange that may carry its offspring into open ground. These roles existed throughout blueberry evolution, before people began measuring the pigments in laboratories. A chemical test can mix blueberry extract with unstable molecules called free radicals. Anthocyanins may donate electrons or hydrogen atoms and reduce the reactions measured in that container. Human digestion creates a longer route. Stomach acidity changes pigment structure. Intestinal enzymes modify attached sugars, and the liver prepares absorbed compounds for transport or removal. Concentration also falls as the material spreads through the body. Any effect on a blood vessel or brain cell depends on the forms that reach that tissue and the way human cells respond. The blueberry's original pigments arose through plant genes, serving ripening, protection, and seed dispersal. Their exact mixture changes across species and cultivars, giving the word blueberry a wide range of the possible chemistry. A blueberry bush growing beside a main forest can look very different from one flowering in southern Georgia. Both may carry blue fruit, yet their height, winter needs, and genetic structure reflect separate histories. Northern highbush blueberry, Vaccinium corymbosum, supplies much of the cultivated fresh market. Wild forms developed around wetlands and moist forest margins across eastern North America. Cultivated bushes commonly reach between 6 and 10 feet tall, with some growing higher when conditions remain favorable. Many northern highbush cultivars need roughly 800 chilling hours or more before their flower buds can develop normally in spring. Large berries and upright branches make these plants well suited to hand-picking and mechanical harvest. A field is usually planted with named cultivars selected for predictable traits. Duke ripens early in many northern regions, while blue crop became widely planted for its reliable yield and adaptability. Growers often plant 2 or more cultivars whose bloom periods overlap, improving cross-pollination while spreading harvest across several weeks. Lowbush blueberry, Vaccinium angustifolium, stays much closer to the ground. Most stems rise only 6 to 24 inches above the soil. Underground rhizomes spread sideways and send up new shoots, allowing one genetic individual to cover a broad patch. A commercial wild blueberry field in Maine or Atlantic Canada may contain hundreds of neighboring clones. Patch boundaries can become visible when leaves turn different shades of red in autumn. Flowering date, fruit flavor, and ripening speed may also shift from one clone to the next. This natural mixture produces smaller berries, with more variation than a field planted. With one highbush cultivar, warmer southeastern climates favor another group. Rabbit-eye blueberry, Vaccinium virgatum, is native to the southeastern United States and can grow more than 10 feet tall. Its leaves tolerate long, hot summers, and established bushes often handle temporary dry conditions better than northern highbush plants. The name rabbit-eye refers to the pink color seen during one stage of ripening, which was compared with the eye of a white rabbit. Many rabbit-eye cultivars need approximately 400 to 700 chilling hours, though the exact requirement varies. Cross-pollination between compatible rabbit-eye cultivars improves seed development and fruit set. Southern highbush blueberries were bred for regions offering even less winter cold. Plant breeders crossed highbush ancestry with lower chill southern species, including Vaccinium darrowi. Some resulting cultivars require only a few hundred chilling hours and can produce fruit during early spring in Florida. Their early harvest reaches markets before many northern bushes have finished flowering. Cold regions created the opposite breeding goal. Half-high blueberries combine northern high-bush ancestry with low-bush traits. Many grow between 2 and 4 feet tall, allowing winter snow to cover much of the plant. Snow traps air and protects branches from temperatures far below freezing. Cultivars such as North Blue were developed for short growing seasons and severe winters. Differences among these groups extend into their chromosomes. A chromosome carries a long package of genetic information. Humans carry 2 sets, while several important blueberries carry additional copies. Northern highbush and common lowbush blueberries are usually tetraploid, meaning they possess 4 chromosome sets. Their cells commonly contain 48 chromosomes. Rabbit-eye blueberries are generally hexaploid, carrying 6 sets and 72 chromosomes. The broader condition of having extra chromosome sets is called polyploidy. During seed formation, matching chromosomes must pair and divide in an orderly way. Crossing plants with 4 sets and 6 sets can produce offspring whose chromosomes pair unevenly. Poor pairing may reduce fertility, even when both parents carry useful visible traits. Breeders must therefore consider chromosome compatibility alongside flavor, climate tolerance, and berry size. Fruit chemistry varies just as widely. Small, low-bush berries contain more skin relative to each gram of fruit. Since most anthocyanins gather in the skin, equal weights of low-bush and large high-bush berries may deliver different pigment concentrations. Ripeness, sunlight, temperature, and cultivar genetics can change the result enough to prevent one fixed wild versus cultivated rule. The plural word blueberries covers a broad biological collection shaped by geography and chromosome history. Those inherited differences supplied breeders with useful raw material, though early growers still had to discover why promising wild bushes often failed after being moved into cultivated ground. For generations, people could recognize an unusually productive blueberry bush without knowing how to reproduce its qualities. Wild fruit was gathered in summer, and promising bushes were sometimes dug from the ground and moved closer to a home or farm. Many transplants weakened over the following years. Garden soil enriched for ordinary crops often had a pH that was too high for blueberry roots. Mineral fertilizers could supply nitrogen in forms the plant used poorly. Moving the bush also disturbed its shallow roots and the mycorrhizal fungi surrounding them. Seed offered another route, though germination was unreliable under the methods commonly used at the time. In a notebook entry dated June 1st, 1907, botanist Frederick Vernon Coville recorded an older account from horticulturist Liberty Hyde Bailey. Bailey had received a bushel of blueberries and failed to raise a single seedling from them. Coville worked for the United States Department of Agriculture and began turning these failures into controlled experiments. His trials ran from roughly 1906 through 1910. Some took place at Arlington Farm in Virginia, an agricultural research site on land later used for the Pentagon. Coville compared blueberry plants grown in alkaline, neutral, and acidic material One successful medium used peat collected beneath mountain laurel, near the Potomac River. Mountain laurel belongs to the same heath family as blueberries and grows naturally in acidic organic ground. By 1910, Colville had shown that blueberry plants required moist soil with strong acidity. A range around pH 4.5 to 4.8 produced healthy growth in his experiments. He also found that seeds germinated more successfully when placed near the surface of an acidic mixture, where they received light and steady moisture. This work supplied a practical growing method, though a breeding program still needed exceptional wild parents. Elizabeth Coleman White provided access to them. White helped manage her family's cranberry farm at Whitesbog in the New Jersey Pine Barrens. Cranberries already grew in the region's acidic sandy soil, and wild highbush blueberries occupied nearby woods and wetlands. Local pickers knew those wild populations one bush at a time. Beginning in 1911, White offered payment for information leading to bushes with unusually large berries. Pickers brought fruit samples to Whitesbog and marked the original plants so they could be found again. Size provided the first filter. Flavor, yield, and the health of the bush determined whether a selection remained useful. One famous wild plant located through this search became known as rhubarb. Its berries were smaller than many modern cultivated blueberries, yet its genes entered early breeding lines and still appear in the ancestry of later cultivars. Colville began making planned crosses in 1911. Workers selected an unopened flower and removed or controlled its own pollen. Pollen from a chosen second plant was applied to the stigma, and the flower was protected from unwanted visitors. Seeds from the resulting berry carried a new combination of traits. Each seedling then had to grow for several years before its fruit could be judged. Most failed to combine every quality a commercial farm needed. The remaining plants were propagated from cuttings, producing genetically matching bushes that preserved the selected traits. Whitesbog harvested its first commercial crop of cultivated highbush blueberries in 1916. Only 6 years had passed since Colville's main soil discovery. Later cultivars descended from this program helped establish blueberry farms in New Jersey and other states. Human selection gradually increased average fruit size. Breeders also favored bushes with dependable yield, useful ripening dates, and berries that separated cleanly from the stem. Cultivation made rows more predictable, while leaving the plant's older biology in place. Blueberry roots still required acidic soil and reliable moisture. Flowers continued depending on pollinators, while ripe fruit remained soft enough to bruise during handling. The crop had moved from uncertain wild transplant to reproducible cultivated variety in little more than a decade. Success created a larger practical challenge. Thousands of productive bushes could now ripen millions of delicate berries that needed to be picked, cooled, and carried far beyond Whitesbog. Commercial success changed the qualities breeders needed from a blueberry. A pleasant flavor still mattered, though fruit also had to survive picking, packing, and travel. Modern breeding programs may evaluate thousands of seedlings created from carefully chosen parents. Several years pass before a new seedling produces enough fruit to judge. Promising plants are then tested across different farms and seasons, making cultivar development a process that can last more than a decade. Breeders measure berry diameter, sweetness, acidity, yield, and resistance to common diseases. Firmness receives special attention because soft cells are easily damaged by pressure. A berry may look unbroken after an impact while bruised tissue develops beneath its skin. The picking scar creates another useful measurement. This small circle forms where a ripe berry separates from its stalk. A dry, narrow scar loses less water and gives fungi a smaller opening than a torn or moist surface. Ripening date can extend a farm's working season. Early cultivars reach market before the main crop, while later plants continue producing after other fields have finished. Harvest method adds further pressure. Workers picking fresh market blueberries can select fully colored fruit and place it gently into containers. Mechanical harvesters move along rows and agitate the bushes with flexible rods. Ripe berries drop onto catching plates before traveling over belts toward a storage bin. Each fall or collision can remove wax and damage cells. Machine harvesting works especially well for firm cultivars and fruit intended for processing. Wild lowbush blueberries are often gathered with hand rakes or mechanical equipment designed for their short growth. Most enter the frozen or processed market. Large cultivated high bush berries supply much of the fresh fruit displayed in clear containers. A harvested blueberry remains alive. Its cells continue respiration, using oxygen to break down stored sugars. This process releases carbon dioxide, water, and heat. Warm fruit respires faster, so removing field heat soon after harvest becomes important. Berries left warm for several hours can lose firmness faster than fruit cooled promptly. Forced air cooling draws refrigerated air through openings in boxes and containers. Fresh blueberries are commonly held near 32 degrees Fahrenheit or 0 degrees Celsius, with high humidity to limit water loss. Cooling slows respiration and softening, while reducing the growth rate of many fungi. It preserves the condition reached on the bush and cannot restore sweetness to fruit picked too early. Freezing slows biological activity much further. Water inside and between cells forms ice crystals. Those crystals can puncture membranes, which explains why thawed berries often become softer and release more juice. Fiber remains, and many vitamins or polyphenols survive well enough for frozen fruit to remain nutritionally useful. The scale supported by these systems is now enormous. United States farms produced approximately 789.5 million pounds of cultivated blueberries in 2024. Their farm value reached about $1.15 billion. Washington, Oregon, and Georgia together supplied almost 2/3 of that cultivated crop. Reported wild production added roughly 90.8 million pounds, with Maine accounting for the recorded national total. Almost all of those wild berries were sold for processing. International growing regions fill the months between domestic harvests. Peru uses seasons south of the equator to ship fruit north during autumn and winter. Mexico can supply berries across an extended period, while Chile's summer overlaps with winter in the United States and Canada. Refrigerated transport keeps fruit cool across journeys lasting thousands of miles. Year-round supply still depends on each plant receiving suitable weather. A warm winter may provide too few chilling hours for a northern cultivar. Early spring heat can open flowers before the final frost. Summer drought strains shallow roots, and extreme heat can soften fruit before harvest. Fungal diseases or reduced pollinator activity create additional pressure. Breeding collections preserve wild plants and older cultivars, whose genes may provide future cold tolerance or disease resistance. A supermarket container therefore joins plant genetics with mechanical handling, controlled temperature, and global geography. After years of breeding and weeks of transport, that long plant journey reaches its boundary when one cooled berry breaks open between the teeth. The first pressure from the teeth flattens the berry and tears its skin. Wax breaks into fragments while pale pulp spreads across the tongue. Thousands of plant cells rupture within a moment, releasing their water and dissolved contents. One level cup of fresh blueberries weighs approximately 148 grams. Water accounts for about 125 grams, or nearly 5/6 of that weight. The serving provides roughly 84 calories and close to 15 grams of naturally occurring sugars. Dietary fiber contributes around 3.5 grams. Smaller amounts include about 14 milligrams of vitamin C and 29 micrograms of vitamin K. Manganese reaches roughly 1/2 of a milligram. This mineral helps enzymes involved in metabolism and the maintenance of connective tissue. The berry also contains organic acids, aroma compounds, and a varied collection of polyphenols. Chewing separates these materials according to their physical location. Anthocyanins leave the pigmented skin and dissolve into saliva or released fruit water. Glucose and fructose escape quickly from broken cells because they are already dissolved. Skin pieces retain cellulose and pectin, which form much of the fruit's structural framework. Humans produce no enzyme that can completely digest cellulose. Some pectin can later be fermented by microbes in the large intestine. Tiny seeds may remain nearly whole because they are small enough to avoid being crushed between the teeth. Saliva moistens the mixture and helps gather loose pieces into a form that can be swallowed. Its pH usually rests near neutral, far less acidic than the stomach. Salivary amylase begins breaking down starch in many foods. Blueberries contain relatively little starch once ripe, so this enzyme makes a modest contribution to their digestion. Muscles in the tongue guide the softened material toward the throat. A wave of contraction called peristalsis carries it down the esophagus. Peristalsis moves food through a coordinated squeeze of circular muscles. This action can continue even when the body is lying down. The swallowed berry reaches the stomach within several seconds. There, muscular contractions fold it into gastric fluid. Hydrochloric acid can lower stomach contents toward a pH between about 1.5 and 3, depending on the meal and timing. The enzyme pepsin begins digesting proteins. A cup of blueberries contains only about 1 gram of protein, leaving physical mixing and acid exposure as the more important stomach processes for this fruit. Acidity begins shifting anthocyanins into chemical forms favored at low pH. The food's original structure still influences how quickly each compound becomes available. Sugars already freed from broken cells mix readily with stomach fluid. Materials enclosed within larger skin fragments take longer to escape. Fiber absorbs water and changes the thickness of the stomach contents. This arrangement is called the food matrix. It describes how nutrients and plant compounds remain packaged within water, cell walls, fiber, and tissue. The intact matrix also spreads release across repeated contractions. Stomach contents leave in small portions through the pylorus, a muscular valve leading to the small intestine. A clear liquid usually moves faster than the same material held in a thick mixture of pulp and fiber. Processing changes that package before the first bite. Juicing breaks cells and removes much of the insoluble skin material. A blueberry puree retains more fiber, though blending has already torn apart most of the fruit. Freeze-drying removes frozen water through sublimation, a process in which ice changes directly into vapor under low pressure. The remaining powder weighs far less than the original berries, so 1 gram represents a concentrated amount of fruit solids. Researchers often use freeze-dried powder when they need participants to receive a measured quantity each day. An extract narrows the composition further. Water, alcohol, or another solvent can separate selected polyphenols from much of the sugar and fiber. The resulting dose may behave differently from whole fruit during digestion. Each form begins with blueberries while delivering a distinct physical and chemical package. Once the swallowed material reaches the stomach, the fruit's role in seed dispersal has ended and human digestion controls its path. Its mixture is often summarized by the word antioxidant, whose familiar meaning has grown much broader than the chemistry it originally described. In chemistry, oxidation begins when electrons move away from one molecule and become available to another. The receiving reaction is called reduction. These paired reactions occur continuously inside every living cell. They allow nutrients from food to be rearranged and their stored energy to become usable. Much of this activity takes place inside mitochondria, the small structures that supply cells with chemical energy. Mitochondria pass electrons through a chain of proteins while using oxygen near the end of the process. A small share of the oxygen can enter reactive forms. One example is superoxide, a molecule containing oxygen and an extra unpaired electron. Another is hydrogen peroxide, which is less reactive than some related molecules, but can still participate in damaging reactions when poorly controlled. Scientists group these and similar compounds under the name reactive oxygen species. Immune cells deliberately produce some reactive molecules while attacking microbes. Exercise temporarily raises their production as working muscles use more oxygen. Ultraviolet light and air pollution can add further sources. Large or poorly controlled amounts may damage membrane fats, alter proteins, or affect genetic material. Smaller amounts perform useful signaling jobs. They can help cells adapt to exercise, respond to infection, and adjust their energy use. Human biology therefore regulates reactive oxygen species instead of trying to eliminate every molecule. Several enzymes carry out this work. Superoxide dismutase converts superoxide into hydrogen peroxide. Catalase can then split hydrogen peroxide into water and oxygen. Glutathione peroxidase uses a sulfur-containing compound called glutathione to reduce peroxides. Cells continually make and recycle these protective materials. Nutrients from food may support or influence the system, while the enzymes provide a built-in line of control. Blood and tissues also contain small molecules such as uric acid and vitamin C that can take part in redox reactions. Their behavior depends on concentration and surroundings. The label antioxidant therefore describes an action observed under particular conditions. Anthocyanins can donate an electron or a hydrogen atom to some reactive molecules. This makes their activity measurable in a laboratory. One widely promoted test was called oxygen radical absorbance capacity, usually shortened to ORAC. Researchers placed a food extract into a solution containing a fluorescent marker. A chemical was added to produce reactive molecules that gradually damaged the marker and reduced its glow. Extracts that preserved the fluorescence for longer received higher ORAC values. Blueberries often scored well because their mixture of polyphenols reacted strongly in that container. Food companies began displaying these numbers as though a higher value predicted greater protection in the human body. The test had never measured digestion, absorption, or disease outcomes. In 2012, the United States Department of Agriculture removed its public ORAC database. The agency explained that antioxidant capacity measured in a test tube could not be treated as proof of a specific effect in people. A digestive system changes nearly every condition used in the assay. Stomach acid alters anthocyanin structure. Intestinal enzymes remove or rearrange attached sugars, and gut microbes can split the pigments into smaller compounds. Only a limited fraction reaches blood in its original form. Once absorbed material enters circulation, it becomes diluted through several liters of blood and distributed among many tissues. The liver modifies compounds, while the kidneys begin removing water-soluble products. Concentrations inside a cell may therefore remain far below those used in laboratory mixtures. Biological effects can still occur through signaling. A small amount of one metabolite may alter enzyme activity. Other products can influence genes involved in inflammation or the cell's own protective defenses. Blueberry compounds may also affect nitric oxide, a short-lived signal that helps blood vessels relax. Gut microbes create additional metabolites whose properties differ from those of the original fruit pigments. This model gives the word antioxidant a precise place within a much larger process. The laboratory reaction remains real, while human outcomes depend on dose, digestion, metabolism, and the response of a particular tissue. Following those conditions now requires returning to the stomach and watching the pigments change as they travel toward the intestine. Within the stomach, blueberry anthocyanins enter one of the most acidic environments in the body. An empty stomach may reach a pH near 1.5, while food can raise that number for a time. Muscular contractions repeatedly mix the berry material with hydrochloric acid. Low pH changes the arrangement of electrons within each pigment molecule. Many anthocyanins favor a positively charged red form called the flavillum ion under strongly acidic conditions. This helps explain why blueberry mixtures often look redder when lemon juice or another acid is added. The exact response depends on which anthocyanin is present and which sugar is attached to it. Some pigment may cross the stomach lining during this early stage. Studies detect anthocyanins in blood surprisingly soon after consumption, sometimes within minutes. The amount absorbed through the stomach remains limited, and most of the berry mixture continues through the pylorus. Small portions then enter the duodenum, the first part of the small intestine. The pancreas releases bicarbonate into this region. Bicarbonate neutralizes much of the arriving stomach acid and protects the intestinal surface. As the contents move onward, pH commonly rises towards 6 or 7. Anthocyanins respond by shifting among several molecular arrangements. At the higher pH, water can add to the flavillum structure and produce a pale form called a hemicatal. The molecule may then open into a chalcone, which often looks yellowish or colorless. Warmth, oxygen, and digestive time encourage further breakdown. Phenolic acids released from these reactions can later be absorbed or passed to microbes. Some forms lose their visible color. Others become less stable and begin opening or breaking into smaller structures. Digestive enzymes also act on the sugar groups attached to polyphenols. The intestinal surface is covered with microscopic projections called villi, which greatly increase the area available for absorption. Each villus carries an outer layer of intestinal cells known as enterocytes. Enzymes along their brush-like surface can remove certain attached sugars before a compound enters the cell. Small molecules may cross through transport proteins built into the membrane. Chemical properties allow others to pass through the fatty membrane more directly. Absorption marks the beginning of further processing. Enterocytes contain enzymes that attach new chemical groups to incoming compounds. Blood from the intestine then travels through the portal vein to the liver, where another large collection of enzymes continues the work. Absorbed products entering portal blood encounter the liver before reaching the rest of the circulation. One process, methylation, attaches a small group made from carbon and hydrogen. Glucuronidation adds glucuronic acid, a water-friendly compound formed from glucose. Sulfation attaches a sulfate group. These changes can alter a metabolite's activity and make it easier to carry in blood. They also help prepare compounds for removal through urine or bile. The kidneys filter many water-soluble products from circulation. Bile carries others from the liver back into the digestive tract. Older absorption studies often searched for only the original anthocyanins found in the fruit. Frequently, less than 1% of a swallowed dose appeared in blood as those unchanged pigments. That narrow result led to the idea that nearly all anthocyanins simply vanished. Modern methods follow a broader chemical family. Researchers can build anthocyanins containing a heavier form of carbon and then track those marked atoms after the molecule changes shape. This technique has revealed modified anthocyanins and smaller breakdown products that earlier tests overlooked. The recovered material may appear across many hours. Original or rapidly modified compounds can peak in the first 1 or 2 hours. Products requiring travel to the colon may rise much later. A blood sample collected at only one moment can therefore miss much of the sequence. Low unchanged absorption still matters because intact pigments rarely reach distant tissue at the concentration present in blueberry skin. The larger biological story includes metabolites created by the intestine, liver, and microbes. Each new form may interact with human cells differently from the molecule made by the plant. Meanwhile, fiber and a considerable share of unabsorbed polyphenols continue through the small intestine. They approach the colon as a chemically altered mixture awaiting enzymes supplied by its dense microbial population. Material leaving the small intestine passes through the ileocecal valve and enters the first part of the colon. Most simple sugars and many vitamins have already been absorbed. Blueberry skin fragments, tiny seeds, and resistant cell walls continue onward. Cellulose remains largely intact because human enzymes cannot split its tightly linked glucose units. Part of the pectin also survives the upper digestive tract. Unabsorbed anthocyanins arrive with other polyphenols and the breakdown products formed earlier. The colon contains one of the densest microbial communities in the body. Hundreds of bacterial species may be present, with their cells greatly outnumbering human cells in each gram of colon contents. The colon's slow mixing movements keep particles near bacteria for many hours. Mucus covering the intestinal wall creates small habitats where different organisms live close to nutrients released from passing fiber. Together, they carry a much wider collection of digestive enzymes than the human genome provides. Some enzymes remove sugar groups still attached to anthocyanins. These enzymes are often called glycosidases because they break bonds involving sugars. Other microbial reactions open the pigment's ring-shaped central structures. A large colored molecule can become several smaller compounds with no blue-purple appearance. Cyanidin-based anthocyanins can produce protocatechuic acid. Malvidin forms can contribute to syringic acid and related metabolites. The exact products depend on the starting pigment and the microbes performing each step. Blueberry fiber enters a separate set of reactions. Bacteria ferment portions of pectin and other carbohydrates, releasing short-chain fatty acids. Acetate is produced in especially large quantities. Butyrate can serve as an important energy source for cells lining the colon. Propionate commonly travels toward the liver, where it enters further metabolism. The amount and balance of these products depend on the entire meal and the microbial community already established. Smaller phenolic acids can cross the colon wall. Blood from this region enters the portal circulation, which carries absorbed material directly to the liver. Liver enzymes may add sulfate or glucuronic acid groups and then release modified metabolites into general circulation. The kidneys eventually filter many water-soluble products into urine. Other compounds enter bile. Bile is made by the liver, stored in the gallbladder, and released into the small intestine to help with fat digestion. A polyphenol metabolite carried in bile can return to the intestinal tract after already passing through the liver. Microbial enzymes may remove the added chemical group and make the compound available for absorption again. This route is called enterohepatic circulation, meaning circulation between the intestine and liver. Repeated movement can extend the time that blueberry-derived material remains detectable. Original pigments may peak in blood during the first few hours. Some microbial products appear 6 hours later or continue rising beyond that point. Food can take a day or longer to travel fully through the colon, allowing further products to emerge gradually. Individual microbial communities make this timing highly variable. One person may carry bacterial species that rapidly open anthocyanin rings. Another may produce more of a particular phenolic acid from the same portion. Habitual fiber intake helps shape which organisms receive a steady food supply. Recent meals can change the material available for fermentation. Medication may reduce or favor certain bacterial groups. and intestinal transit time changes the hours available for each reaction. Human genetics also influences digestion and immune conditions around the microbes. Researchers have tested whether regular blueberry intake changes the abundance of particular organisms. Some studies report shifts in bacteria associated with fiber fermentation, or changes in microbial metabolites. Other trials find limited effects, and the named groups do not remain consistent across every population. No single bacterial pattern can currently be described as a universal blueberry microbiome. The dependable finding lies in the transformation itself. Microbial enzymes finish reactions that human digestive enzymes cannot perform. Their smaller products can cross into the body and reach tissues far from the colon. A serving of blueberries, therefore, produces a chemical profile shaped jointly by fruit composition and the organisms already living in the intestine. Once these metabolites pass through the liver and enter general circulation, they meet the single-cell thick lining spread across the entire network of blood vessels. After leaving the liver, blueberry metabolites enter veins carrying blood toward the heart. A heartbeat then sends them through arteries and into smaller branches throughout the body. Some travel freely in the watery plasma. Others attach loosely to albumin, the most abundant protein in blood, which can carry many small molecules. Every route brings them close to the endothelium. This is the single-cell thick lining covering the inner surface of blood vessels. An endothelial cell is extremely flat, allowing a smooth layer to extend from the largest arteries into microscopic capillaries. Its position gives it continuous contact with flowing blood. These cells sense pressure and the friction created as blood moves across them. They respond to hormones, immune signals, and nutrients. Their activity helps control clotting and the movement of immune cells through vessel walls. Changes in endothelial signaling also adjust vessel diameter from moment to moment. Nitric oxide provides one of the main widening signals. Endothelial cells make it from the amino acid arginine through an enzyme called endothelial nitric oxide synthase. The gas lasts only briefly and passes into nearby smooth muscle cells wrapped around the vessel. There it activates another enzyme that raises a messenger molecule called cyclic GMP. This messenger lowers tension within the muscle. As the circular muscle relaxes, the vessel widens and resistance to blood flow falls. Nitric oxide can be lost when it reacts with excessive superoxide, one of the reactive oxygen species described earlier. Inflammatory signals may also reduce the activity of the enzyme that produces it. Blueberry metabolites could influence several points in this system. Laboratory studies suggest that some may support nitric oxide production or reduce enzymes that generate superoxide. Others may change signals controlling inflammation and the cell's own protective enzymes. Concentrations in blood remain low, so researchers focus on changes in cellular communication at those measured levels. A method called flow-mediated dilation allows this communication to be measured in people. The participant rests while an ultrasound probe records the diameter of the brachial artery in the upper arm. A pressure cuff is inflated for about 5 minutes, temporarily reducing blood flow below it. Releasing the cuff sends blood rapidly through the artery. The increased friction encourages healthy endothelial cells to release nitric oxide. Ultrasound then measures how much the artery widens, usually as a percentage of its resting diameter. A larger response generally indicates better endothelial function. Even a change of 1 percentage point can be detectable under controlled conditions. Controlled blueberry trials have used whole berries, drinks, or freeze-dried powder equal to 1 or 2 cups of fruit. Some record modest improvements in flow-mediated dilation within a few hours. Other interventions provide a daily portion for several weeks and measure whether the response persists. Blood pressure studies produce a less uniform picture. Trials involving adults with elevated cardiovascular risk sometimes report reductions of a few millimeters of mercury. Several studies detect little change or no clear difference from a comparison group. Starting blood pressure matters because a person already within a healthy range has less room for a measurable reduction. Dose, cultivar, and processing can alter the compounds delivered. Medication use and the length of the intervention add further variation. Large observational studies offer evidence across longer periods. Researchers may compare reported anthocyanin intake among tens of thousands of participants with later rates of high blood pressure or cardiovascular disease. People who regularly eat berries may also differ in exercise, smoking, income, or the rest of their diet. Statistical methods can adjust for recorded differences while leaving unmeasured factors behind. Randomized trials provide a stronger test of cause because researchers assign the intervention. Their smaller size and shorter duration limit what they can reveal about disease developing over decades. Current evidence, therefore, supports a plausible pathway from blueberry metabolites to endothelial signaling, with modest effects appearing under some conditions. The fruit remains a food and cannot replace medication or medical care. Vessel relaxation forms one measurable part of the body's response. At the same time, sugars from the berry are entering circulation and calling on insulin, metabolic tissues, and immune signals to coordinate the rest of the meal. While polyphenol metabolites circulate at low concentrations, the berry's sugars follow a more direct route. Ripe blueberries contain mainly glucose and fructose, with smaller amounts of sucrose in many cultivars. Enzymes along the small intestine split sucrose into its 2 component sugars. Glucose enters intestinal cells largely through a transport protein that moves it together with sodium. Fructose uses a different transporter called GLUT5. Both then pass into portal blood, traveling toward the liver. The liver removes much of the arriving fructose and converts its carbon into usable metabolic materials. Part may support immediate energy needs or contribute to stored liver glycogen. Glucose continues more widely through circulation, raising blood glucose after the meal. Whole fruit structure affects the speed of this process. Some sugar remains enclosed within partly broken plant cells after chewing. Pectin absorbs water and increases the thickness of material moving through the intestine. Fiber also slows physical mixing and changes how quickly sugars reach the intestinal surface. A filtered blueberry drink has already lost much of this structure, allowing dissolved sugars to become available more rapidly. Blended puree falls somewhere between intact fruit and clear juice because it retains fiber while breaking many cell walls. Rising blood glucose is detected by beta cells in the pancreas. Glucose entering these cells is used to produce chemical energy. That change closes channels in the cell membrane and allows calcium to move inward. Calcium triggers small packages of insulin to fuse with the membrane and release their contents into blood. Insulin travels to tissues carrying receptors shaped to recognize it. In muscle and fat cells, the signal helps move GLUT4 transporters toward the cell surface. These proteins allow more glucose to enter. The liver responds by increasing storage of glucose as glycogen and adjusting its own glucose production. Insulin sensitivity describes how strongly tissues respond to a given insulin concentration. High sensitivity allows a smaller signal to produce an effective response. Reduced sensitivity requires the pancreas to release more insulin to manage the same rise in glucose. Blueberry polyphenols and their metabolites may influence parts of this system. Cell experiments suggest effects on insulin signaling proteins and enzymes involved in carbohydrate digestion. Some studies also examine pathways controlling glucose transport or energy use within muscle. The concentrations used in laboratory cells can exceed those reached after eating ordinary fruit. Animal research permits direct examination of liver and muscle tissue, though species differences limit its transfer to humans. Controlled human trials provide a closer test. Some have given adults freeze-dried blueberry powder for several weeks, often in amounts representing 1 or 2 cups of fresh berries per day. Selected studies involving participants with reduced insulin sensitivity report modest improvement in the way tissues respond to insulin. Other trials find smaller changes or no clear difference. Starting health, dose, study length, and the comparison food all influence the result. The blueberries' sugars and energy remain part of the meal throughout these responses. Inflammation adds another connected system. A short inflammatory response helps contain infection and begin tissue repair. Immune cells release signals that change nearby blood vessels and recruit additional defenses. Problems can develop when inflammatory signaling remains elevated or poorly controlled over long periods. Researchers often measure C-reactive protein, which the liver produces after receiving certain immune signals. Lower or higher C-reactive protein provides a broad indication of inflammation without identifying its exact source. Blueberry trials sometimes report changes in this marker or in individual signaling molecules. Results differ enough to prevent one uniform conclusion. Each research design answers a different kind of question. Cell culture can reveal a possible molecular pathway. Animal studies show how several organs respond together under controlled conditions. Observational research finds associations among people choosing their own diets. Randomized trials assign an intervention, creating stronger evidence of cause while often remaining limited in size or duration. Across these levels, blueberries appear capable of interacting with metabolic and inflammatory signaling. The expected effects are modest and depend on the food form and the person being studied. Blood vessels and metabolic tissues provide measurable outcomes, while the most familiar promise concerns the organ relying continuously on both glucose and circulation. The brain receives glucose and oxygen through blood vessels that branch into an extremely dense network of capillaries. An adult brain usually represents close to 2% of total body weight. At rest, it uses roughly 1/5 of the body's oxygen consumption. Neurons store very little fuel, so circulation must keep delivering glucose and oxygen from moment to moment. Even brief changes in supply can affect how quickly these cells communicate. Long-term vascular health also influences the condition of brain tissue across decades. This dependence creates one plausible route for blueberry metabolites. Endothelial signaling helps adjust cerebral blood flow, meaning the blood moving through the brain. Nitric oxide can relax small arteries and alter the amount reaching an active region. A modest improvement in vessel function could therefore change the environment surrounding neurons without acting directly on the neurons themselves. Other pathways involve inflammation and cellular stress responses. Polyphenol metabolites may influence enzymes or genes that regulate these systems. Laboratory experiments also examine synaptic plasticity. A synapse is the narrow connection where one neuron passes a signal to another. Plasticity describes the ability of that connection to strengthen or weaken with experience. This process supports learning and memory. Some cell and animal studies report changes in signaling proteins involved in plasticity after exposure to berry compounds. Doses and metabolism in those experiments can differ greatly from ordinary human eating. The blood-brain barrier adds another filter. Tightly joined cells around brain blood vessels control which compounds enter surrounding tissue. Small blueberry-derived metabolites may cross under certain conditions. Intact anthocyanins usually circulate at very low concentrations, making a large accumulation of original pigment in the brain unlikely. Human trials examine the combined result through cognitive testing. Children in some acute studies consume a wild blueberry drink and complete tasks several hours later. Researchers may ask them to remember a word list, ignore distracting information, or switch between two rules. Other experiments follow adults for 8 to 12 weeks, while providing daily whole berries or freeze-dried powder. Older participants may complete tests of verbal memory and processing speed. Studies involving people with mild cognitive concerns sometimes use paired associate learning, where two related items must be remembered together. Selected trials report improvement in one or more measures. A memory score may rise while attention remains unchanged. Another study may detect faster responses alongside a small increase in errors. Several experiments find uncertain results after researchers account for the number of tests performed. Participant numbers often remain below 100, and some early studies include far fewer. A small trial can identify a useful signal while producing an estimate that may change in a larger group. Intervention length creates another limit. One drink can test an effect appearing over several hours. A 3-month trial may reveal short-term adaptation. Neither design can establish what happens over 20 or 30 years. Cultivar and preparation add further variation. Wild blueberry powder may contain a different anthocyanin mixture from cultivated high-bush fruit. Juice delivers dissolved compounds with less intact fiber. Extracts concentrate selected molecules, while whole berries preserve the original food matrix. Fresh and frozen fruit can both retain useful polyphenols. Ice crystals break cell membranes during freezing, so thawed berries become softer and release more juice. That texture change does not remove all fiber or pigment. Storage time and temperature still influence vitamin and polyphenol levels. Concentrated supplements require separate evidence, because their dose and composition may differ from food. Current human research therefore supports plausible pathways and some encouraging cognitive results. The findings vary across age groups, tests, and study designs. Improvement on one task demonstrates a change in that measured ability during the study. It cannot establish prevention of dementia or reversal of neurological disease. Blueberries fit most securely as one fruit within a broader eating pattern. Their metabolites may interact with circulation and cell signaling, while the size and long-term importance of those effects remain under investigation. That measured uncertainty completes the evidence-based path from swallowed pigment to human tissue. The ordinary berry can now be viewed again with its plant history and bodily transformations held together in one continuous journey. A blueberry can now rest in the hand with every part of its journey still visible. Its dusty blue surface comes from microscopic wax structures that scatter blue and ultraviolet light. Beneath that bloom, the skin holds most of the dark anthocyanin pigments and a substantial share of the fruit's fiber. The pale interior is mostly water, with dissolved sugars, organic acids, and thin-walled cells that collapse easily between the teeth. At one end, the small five-pointed crown preserves the calyx, a structure left behind by the flower. Inside, dozens of tiny seeds carry evidence that pollen once reached the flower and fertilization followed. That visible fruit began in soil acidic enough to challenge many ordinary crop plants. Fungal partners called ericoid mycorrhizae extended the reach of its delicate roots through organic soil. Months earlier, cold winter temperatures had allowed dormant buds to complete their chilling requirement. Warm spring weather then encouraged the flowers to open. A visiting bee grasped a flower and vibrated its flight muscles, shaking pollen through narrow openings in the anthers. When enough ovules were fertilized, developing seeds produced hormonal signals that encouraged the surrounding ovary to swell. During ripening, its cell walls loosened, acidity declined, and sugar became more noticeable. Anthocyanins accumulated in the skin as green chlorophyll faded. The growing color helped make ripe fruit visible to birds and mammals, capable of carrying seeds away. Human cultivation entered this older biological chain only recently. During experiments from about 1906 to 1910, USDA botanist Frederick Coville learned that high-bush blueberries required moist soil near pH 4.5 to 4.8. Elizabeth Coleman White added detailed knowledge of New Jersey's wild bushes and acidic cranberry land. Local pickers marked plants with unusually large fruit, giving the breeding program useful variation to preserve and combine. Their work respected requirements already written into blueberry biology. Cultivated plants continued to depend on suitable acidity and well-timed winter cold. Their flowers still needed effective pollen transfer, often supplied by managed honeybees or wild bees. Refrigeration stretched the distance and time between field and table. Rapid cooling slowed respiration, fungal growth, and water loss in harvested berries. The living fruit continued using stored energy, however, and no cold room could reverse bruising or restore quality lost through early picking. A bite began a different series of transformations. Chewing cracked the waxy cuticle and tore pigment-rich skin into fragments. Stomach acid temporarily favored reddish forms of some anthocyanins. Conditions changed again in the small intestine, where bicarbonate reduced acidity and enzymes began rearranging the pigments. Only a small fraction entered the blood as the exact molecules once present in the skin. Intestinal cells and the liver attached chemical groups that changed how the compounds traveled and how quickly they could be removed. Material that escaped absorption reached the colon alongside intact fiber and broken cell walls. Bacterial enzymes separated sugars from polyphenols and opened their ring-shaped structures. Smaller phenolic compounds could then cross the colon wall, pass through the liver, and enter circulation in altered forms. Possible effects on blood vessel relaxation, glucose handling, inflammation, or cognition would emerge through these metabolites and the signals they influence. Human trials sometimes measure modest changes, while other studies find uncertain or negligible differences. The word antioxidant accurately describes an ability to take part in certain electron transfer reactions. It cannot, by itself, explain digestion, metabolism, or the response of living tissue. A whole blueberry carries a more complete account. Its existence links an acid-tolerant shrub with soil fungi and a vibrating bee. It also links careful growers with refrigerated transport, gut microbes, and human cells responding to chemical signals. Each participant changes what reaches the next. The fruit remains small enough to disappear in a single bite, yet its simplicity has quietly fallen away. The chemical object entering the mouth differs from the one that ripened on the bush. Hours later, its compounds differ again. Even after those changes, the final metabolites still carry traces of a pathway that began with acidic ground, a spring flower, transferred pollen, and a berry slowly darkening beneath its pale wax.